Hey, welcome back. You guys have been spending a lot of time learning about DNA, RNA, coding RNA, non-coding RNA, complementarity, lots of interesting concepts. And you're wondering, why do I need to know all this? And you will soon find out, because you can't understand Cas9 as a protein unless you understand non-coding RNA, because it is not a functional protein without using non-coding RNA. And this is very unique. You don't see a lot of proteins like this, but that's kind of what makes it a cool protein. So good for you for understanding all this, because now it's going to all come together and it's going to make sense. What I'd like you to do is go through this to try to understand how a bacteria arms itself against the attack of bacteriophage, attack of viruses. And then after that, if you have the Cas9 kit, the making the cut kit, you can work on that part of it. And then we are going to really delve into the Cas9 protein and understand how it does its thing. But right now we're understanding the big picture, how the system works, and then we'll understand the actual structure function of the Cas9. All right, so without further ado, let's look at the adaptive immunity system of the bacteria, the CRISPR system. So this is a graphic of a bacteria being under attack by viruses known as bacteriophages. Bacteria are constantly under attack by viruses, and over the millennia they have evolved several systems to help protect them against these attacks, and one of those systems happens to be the CRISPR-Cas system. So we are going to go through how this happens so that you can understand the part that non-coding RNA plays in this system, in this system. Okay, so in this case, we have these different viruses, and we'll talk about these two later. But let's just look at the purple virus. So the purple virus is getting ready to attack and basically inject its genetic material into the bacteria, hoping to produce lots of more bacteria to go on for further infection. Right? So what happens is it might do that. It might take over the bacteria, kill the bacteria, make lots more viruses, and move on. But but sometimes the bacteria survives that attack. And when it does, it can take a snapshot of that virus so that it can know when that specific virus comes back and be ready for it and be able to then cleave its genetic material and basically kill the virus. So what it does is, as the genetic material is inserted into the bacterium, it takes a snippet of that and inserts it into the bacterial genome in what is known as the CRISPR locus. It's a part of the genome that's whole job is to keep track of the viruses that have been trying to invade it. All right, and so what it does is it inserts that genetic material. This purple space section here is the sequence. It's part of the sequence of that virus. So it will always know when that virus comes back, that type of virus comes back. Okay, now how does it do that? It does that in a very particular way. We don't need to know about all of the machinery that's involved in cleaving that and inserting it into the genome. But what you have to know is that it can take any sequence, but what it wants to do is it wants to bookmark that sequence, and it wants to know where in that genome it is so that it can readily check for it later, okay? And so what it does is it uses something called a PAM site. So the section that's going to be cleaved from that virus in the virus is called a protospacer. They've called it a protospacer. And the PAM site is the protospacer adjacent motif. It's a little sequence, and it changes depending on what Cas system you're talking about. In Cas9, it is NGG, so it's any nucleotide followed by a guanine and a guanine, NGG. And so it goes through, finds an NGG in that genome, and then cleaves the section right next to it. It doesn't take the NGG, it binds to that and takes the section next to it. And that'll be important later. And then it sticks it into the bacterial genome. Okay, now, how does that, and you can see here, so these are previous infections by the blue virus and by the tan virus, okay? So it just keeps adding this to this, what we call the Crispr array. And then later, it can go out and check for viruses that are coming in to see if it matches any of these. And I'll show you how it does that. Okay, so I told you that some RNA is coding RNA and codes for proteins, and some RNA is non-coding RNA and is functional all by itself as RNA. And it does so by forming hairpins and forming twists, forming tertiary structure that allows it to work, in some cases, very similar to proteins, okay? So what we have to do is understand how this part of the genome is transcribed and what is non-coding RNA and what is coding RNA. And so it turns out that what happens is this whole thing is transcribed, and then it's broken up into pieces, and some of it is translated into protein. So this white piece is translated into protein, and that is the Cas protein. It's the nuclease that cuts the DNA once the sequence is found, and that is going to be indicated by a little white blob like that, okay? This over here, orange stuff, is called tracer RNA. It's called tracer because it's trans-activating CRISPR RNA. And this over here is the CRISPR RNA. And it is, like I said, consists of all these pieces of viral DNA that now keep getting passed on to by replication. So it's sequence that originated in viruses. And they're separated by these black pieces, which are the clustered, regularly interspaced, short palindromic repeats. And that is how CRISPR was originally discovered, was because they saw what these repeats that they kept showing up. What they didn't realize is the spacer, they just called this spacer material because they just didn't know what it was for. That's the really important part, right? That's the snapshot of the virus that we have to use to find incoming viruses or to find sequence, right? Okay, so the cool thing about these repeats is, like palindromes that we discovered, they can fold and bind to themselves. They could form hairpins, and in other CRISPR systems they actually do that. In this CRISPR system, because it evolved slightly differently, it uses a different section to bind to. So what happens is these get transcribed, they get cleaved by different machinery, and so a piece of repeat and a piece of viral sequence is selected and is transcribed, and this orange piece here, the tracer RNA, is transcribed. And what happens is they work as non-coding RNA. And this repeat happens to be complementary to part of the tracer. And so that is able to bind to that. And the rest of the tracer actually is complementary to itself. It forms hairpins, it forms stem loops, and it is much longer. I'll show you a picture of it, but a lot of the structures that we have don't even show it all because it's very elaborate. It's got like 3 stem loops on it. And that's important for reasons that we will talk about later. So these 2 pieces of RNA that have bound together at this repeat then get packaged into the Cas9 protein. The Cas9 protein is nothing without this, what we call a guide RNA, because it can't bind to DNA unless it has this. In fact, Cas9 grabs onto this, especially this tracer, in a very special way that puts it into a conformation that makes it active. Okay, so this is critical. So this is how non-coding RNA activates the protein Cas9. And here's a bigger version of it so that you can see what that looks like. Okay, now what these things are is Cas9 surveillance complexes. They are what go out, and they're made by the bacteria, and they go out and hang around and look for incoming viruses. And if the purple sequence, the purple viral DNA or RNA shows up, it will bind to that and activate Cas9 to cleave it and break it apart. Okay, destroy that genome. Okay, does that make sense? All right, let's stop there, and then we will go and talk about how this compares to the actual structure.